Okay, I'll go ahead and get started. So, good afternoon, everyone. So, last week we saw the best laboratory practices, and so this week we'll talk a little bit about kidneys. Okay. So, the topic that we are going to discuss today is on kidney structure and function. It's on mammalian kidney. And what we are going to discuss is on about adult kidneys. They are called as metanephric kidneys. So it's a very broad topic, and so I touched on few things, few aspects. So the objective is for all of you who are new to kidney research to get familiarized with some of the terms. I know Mohammed, he has lots of experience working with kidney. Probably he's more familiar with what I'm going to discuss today. But please add in if you think something is missing. Okay, yeah. And also the kidney experts, please fill in if anything is missing in my talk. So the thing is, once you get familiarized with some of the term, it will set you in a kind of a framework for the lectures in the coming weeks. Because in the next few weeks, we are going to discuss about the kidney development and renal physiology, and also about kidney diseases. So the little bit familiarity about the kidney structure and function would help you very much in connecting when you are attending these presentations. Okay, so okay. So the urinary system is the main excretory system, and it has the following structures: the kidneys that produce urine, and the urine that is produced by the kidney are conveyed or transferred through the ureter, and it is stored in the urinary bladder, and then it leaves the body through urethra. So in the excretory system, the kidneys are like a very like a workhorse, working hard, filtering day in and day out the blood and removes the waste products. So the main functions of the kidney is to maintain the body homeostasis, and this is done by regulating the composition and volume of the body fluids, by maintaining the water and electrolyte balance, and also the acid-base balance. And it forms urine and thereby enables the excretion of waste products. So kidneys receive like 25% of the cardiac output, and it filters like almost like 180 liters per day. And then it reabsorbs most of them, and in order to give like 1.5 liter of acidic urine. So it does lots of filtration, reabsorption, and all those things. So the next important function is it regulates the various properties of the blood, the ionic composition of the blood by regulating the concentration of several ions. And also, importantly, the pH by excreting the hydrogen ion and conserving the bicarbonate ion. And maintains the osmolarity by regulating the loss of water and solutes in the urine, and blood volume by conserving or eliminating the water, and blood pressure. Thereby, it regulates the blood pressure. And also the kidneys, they secrete the enzyme, the renin that our lab is working on, and it's a main component of the renin-angiotensin system, which regulates the blood pressure. And also the proximal tubules in the kidneys, they also produce synthesize glucose molecule. In addition, the kidneys are the source of vitamin D and also the erythropoietin. So the erythropoietin, it stimulates and produces red blood cells, and through whenever, and it's very helpful. Like during hypoxic condition, the EPO is expressed and produce red blood cells. So having seen the main important functions of the kidney, now let's go through the cross structure of the kidney. So when you take a longitudinal kidney or a cross section, these zones are demarcated. So it is surrounded by membranous capsule, and then has the outer stripe is called as the cortex of the kidney. And then this is followed by the outer medulla, which is the first outer stripe of the outer medulla, and then the inner stripe of the outer medulla. And this is the inner medulla. And the tip of the kidney is called papilla. So the mouse kidneys, it has a single papilla, whereas the human kidney has several, more papilla. So the renal cortex is enriched with what is called as renal corpuscles. It's a part of the nephron. And the renal corpuscle here, like if you see having the red, the capillaries that are highlighted in red, they are glomeruli and surrounded by the Bowman's capsule. The entire structure is called as a renal corpuscle. And the filtration of the blood starts with the renal corpuscle, and the renal corpuscle and the proximal tubules are enriched in the cortex region. And also the cortex is also supplied with blood vessels, as you see here. And the next comes the renal medulla, the next layer. Okay, so we have like an outer medulla and the inner medulla. So the medulla is, like a pyramid-like structure, which has long tubules, which has loop of Henle, and it is also enriched by vasorecta. Vasorecta is the capillary bed that interacts with the tubules, and it's important in reabsorption process. So in the medulla, there is a lot of absorption, reabsorption, and secretion, and thereby we get the urine. So the renal pelvis is the last, the final area where the filtrate formed or drained by the nephron are collected here, and then it is excreted out. Okay, the urine comes out of the papilla and temporarily in the pelvis region, and then it goes to the bladder through the ureter. So the important structural components of the kidney are like, as I said, the nephron and the collecting duct epithelium, and the renal interstitium. So the space between all these renal tubules is, filled with interstitial cells, and the renal interstitium is the main part of the renal parenchyma. And the renal interstitium also serves as an extracellular matrix, and they have lots of cells like fibroblasts, macrophages, immune cells, dendritic cells, etc. And then the kidney is a highly vascularized organ because it's very important for removing the waste products and also reabsorption. And also, the kidneys, they have renal innervation is also the main component. So the kidneys, they're innervated and they receive nerves from the renal plexus, and they have the afferent and efferent nerves. And the sympathetic part of the renal innervation is important for the blood pressure regulation and also for as a baroreceptor mechanism. So in today's presentation, I'll be focusing more on this one, the nephron and the collecting duct epithelium. So here the urine is, the blood is filtered and the urine is formed. And so we will see how each part of this whole complex contributes in concentrating the urine. So nephron, like these are the structural and functional unit of the kidney. And we have like each kidney contains like around 1 million or 1.3 million nephrons, and it depends like also if you're born with a low body weight or like that, or a premature birth, then the nephron number would be slightly low. and that is, that condition is prone for kidney diseases and hypertension. Yeah.
That number is for humans.
Yeah. Human. In the mouse, it is like around, if I remember, like 12,000 or 10,000 to 12,000.
And I think one of the presentations on Research Day pointed out it's like every kilogram body weight at birth was like 250,000 less nephrons. So for that early birth, significant drops in nephron count.
And that's an important point. Like when we are born, the nephron numbers are defined for human. Whereas for a mouse and all, there is after birth, for initially like around 3 to 5 days, there is nephrogenesis to continue. So we are born with a defined number of nephrons, especially in humans. So the nephron consists, it starts with the renal corpuscle. Okay. So the renal corpuscle has a glomerulus, and this is surrounded by a Bowman's capsule. And the rest of the part is a tubule system. I'll go to the different segments in the next slides. So the renal, the nephron tubule, we have different segments. And the first is the proximal tubule, and PCT is a proximal convoluted tubule, and there is also proximal straight tubule. And then like as I said before, the proximal tubule, the cortex is enriched with the proximal tubule. And then it goes further deeper to the medulla region, and in the medulla we have the loop of Henle, and we have a thick segment of the thick segment, and this is a thin segment. Since this is going down to the medulla, we call it as a descending loop of Henle. And then it goes up to the cortex, and this region is the ascending loop. Again, here we have a thick ascending loop and thin ascending loop. And then this part is the distal convoluted tubule. And then there is a connecting tubule also, and which connects the nephron epithelium with another structure called collecting duct. Okay. So if you look at like, though it is a continuous tubule structure, in each segment, the cell type is different. And this is very important, because each segment has a different function in concentrating the urine. So the next is we have two types of nephrons. One is a cortical nephron, as the term indicates, the cortex, they are present mostly in the cortex. And the next one is the juxtamedullary nephron. And the juxtamedullary nephron, they play an important role in concentrating the urine. So this is because how it happens is this the renal interstitium here, it's a hypertonic or a hyperosmolarity. And and also it has a salt gradient. The more you go, the deeper the salt concentration is high. And this gradient helps in concentrating the urine by reabsorption. So there are four main steps for the formation of urine. And first, it's a glomerular filtration. And the glomerular filtration occurs in the renal corpuscle. And then the tubular reabsorption. Once the blood is filtered in the renal corpuscle, it goes to the proximal tubule, and where a lot of water is reabsorbed. And then when all along the tubular system, the tubular secretion also happens. So since we have this vas recta, the vasculature and the tubule, they interact. Okay, and even from the blood, like toxic any base substance or drugs, material, and those things are all secreted inside the tubule, and together they are excreted. And the final step is urine excretion. So of course, let's get into the renal corpuscle, like how the structures of the renal corpuscle helps in the blood filtration or glomerular filtration. So the important component of the renal corpuscle first is the glomerulus. And the glomerulus comprises the capillary tuft, okay, and it has a capillary endothelium. And then we have this Bowman capsule, and the Bowman capsule has the parietal epithelium, and also we have constructed podocytes. Okay, they have in close contact with the capillary tuft. And then we have a glomerular basement membrane, you see here. So is this pointer okay, or you want me to change to laser thing? Can you see? This is okay? Yeah. So yeah, glomerular basement membrane we have. So next we see about the glomerulus. So as I mentioned before, the capillary tuft inside the renal corpuscle that is called as glomerulus. But in everyday, day to day, we use say the whole structure as glomerulus actually, but there is a demarcation. Okay? The glomerulus means like it's a capillary tuft inside. So the glomerulus is composed of an afferent arteriole where the blood enters the capillaries, and then the filtered blood gets out of the efferent arteriole. So we have the mesangial cells. You see the blue ones, these are the mesangial cells, and they provide the structural support between the glomerular capillaries. And once the blood enters and it is filtered into the Bowman space, and the filtrate is called the ultrafiltrate. The ultrafiltrate goes further down to the proximal tubule, and all this concentration process occurs to get as a final urine. So, like here, in the renal corpuscle. And this is called as vascular pole, where we have the afferent arteriole and efferent arteriole, where the vasculature is there. And where the glomerulus connects, or the renal corpuscle connects to the tubule, that area, that end is called as urinary pole, okay, or tubular pole, or this is a vascular pole. So the important aspect of this renal corpuscle is the glomerular filtration barrier. So you'll see how this glomerular filtration barrier helps in filtration and also in regulating GFR and everything in the physiology lecture. But I'll touch on the structural components that are essential for regulating the filtration. So the glomerular filtration barrier has few components. The glomerular endothelium. So okay, this structure is blown up here. The capillary with the endothelium and the podocyte is blown up here, and this constitutes the glomerular filtration barrier. So first we have the endothelium, and this endothelium is fenestrated. Okay. So it's not continuous, it has gaps, okay? And importantly, the pore size is 70 to 90 nanometer, and this enables a size selective filtration. And then we have the glomerular basement membrane, which is also like ion selective barrier. And we have the podocytes. The podocyte and the unique structures, they have the foot-like processes, okay? And that's what we see here. And these foot-like processes are called as pedicels. And we have the slits in between the pedicels, which is called as a slit diaphragm. And this also acts as a size selective barrier, okay? So when there is a defects in the glomerular basement membrane or in the filtration barrier, what happens is all these large molecules will come out like proteins and causes protein in the urea, which is called as proteinuria. So this scanning EM of that GBM or the glomerular filtration barrier that we saw. So here you can see all the podocyte with all these pedicel, the finger-like process, and here, like you can see the capillary and the podocyte next to it. And this picture also, you can see the glomerular basement membrane and the fenestrated endothelium and the podocytes with all these processes. So this filtration barrier, it acts as a kind of a molecular seal. And this helps in, as I said, it allows only the small molecules from the blood to the urine up to 4 nanometers diameter, while limiting the passage of macromolecules such as albumin and other. And the renal corpuscle also has mesangial cells, and we have two types of mesangial cells: the intraglomerular, the name itself shows that it's within the glomerulus, and also the extraglomerular. And also the juxtaglomerular cells, it's also a part of glomerulus. They're, like it lines the afferent arteriole, and juxtaglomerular cells are the cells that produce renin. And I just wanted to introduce this structure also, the macula densa. Actually, it's a part of the distal tubule, but the region of the distal tubule that comes into contact with the afferent arteriole is called as macula densa. So the macula densa and the glomerular mesangium and the renin cells, they all communicate with each other when there is, the salt concentration in the filtrate is low or high, and accordingly the renin is secreted, and then the blood pressure is regulated. So now we finish the renal corpuscle, and then next we discuss about the tubular epithelium. So we already know the different segments of the tubular epithelium, and so they are like a single layer epithelial structure. And if you see, the cell types are heterogeneous; they are not look similar, right? And I mentioned it before, like this heterogeneity is important for fulfills the function of each segment. So, for example, the basolateral membrane of the PCT, the proximal convoluted tubule, the distal tubule, and tall is the thick acidic loop of Henle. So, if you see, they're all enriched with mitochondria, right? Whereas the collecting duct and the thin descending loop of Henle, we don't see that much of mitochondria. This is because, like the mitochondria, they supply a lot of energy for these cell types, because they have to, push the solutes and everything against the concentration gradient. So they need a lot of energy. So they have, so that's why they have abundant mitochondria. Whereas these two cell types, they also take part in filtration and absorption, but they do passively. So they don't need that much of metabolic activity in these cells. So the proximal convoluted tubule. So once we get the ultrafiltrate, the first part is they go to the proximal tubule, right? And the proximal tubule, like they have like how to say, the cuboidal cells. So the other important thing is this brush border structure. If you see in the top, there are a lot of microvilli, and they have it's a thick layer, and it's a characteristic nature of the proximal tubule. And this why they are enriched with this much, because this helps in reabsorption and also in secretion. So the proximal tubule cells also, they are having Golgi with ER, endoplasmic reticulum, and vacuoles, and this helps in synthesizing and secreting a lot of molecules. And again, like as I mentioned before, they have numerous mitochondria which supply energy for the active transport of water and ions. So the proximal tubule is the region where like maximum amount of water is reabsorbed. Like it's almost like 66% of water And also the bicarbonate ions, like 80% of them are reabsorbed. And this is very, very important because the bicarbonate comes out and the hydrogen ions gets in, and which is how to say, important for the acid balance, acid base balance. So if you see the filtrate at this stage, they are isoosmotic. That means the salt content is almost equivalent to the outside the plasma, which is like around 300 milliosmol. Okay, so the ultrafiltrate is like around 300 milliosmol. Whereas when it comes out as a urine, it is like around like 850 to 1000. It could be a little bit, how to say, the range that is there. So it starts with 300 but ends up with 1000. So that means lots of reabsorption and concentration is happening all along the tubular epithelium. So in the proximal tubule, a lot of water gets out. Okay. And then there is this the bicarbonate and hydrogen exchange, and also it's a place they synthesize the glucose as well, and it's also like a synthesis and also reabsorption of glucose happens in the proximal tubule. And organic acids and bases and the drugs, they are secreted into it, and this comes from the vasorecta, that is the vasculature that is serving the proximal tubule. And next is the loop of Henle. Once the filtrate passes here and loses water, then it goes to the loop of Henle. So the loop of Henle is very, very important in concentrating the urine. Like I said before, because of the hyperosmotic interstitial fluid, lot of reabsorption happens in this, the concentration happens in the loop of Henle region. And we have like, and this is very important because it regulates the fluid volume and thereby the blood pressure, and also the acid balance, acid-base balance with the bicarbonate reabsorption. So the ability of the kidney to concentrate the urine depends upon the length of the loop of Henle. For example, like the aquatic organisms like fish or any other aquatic animals that so they don't need to conserve a lot of water, right? So in those there the loop of Henle is shorter, okay, or even fishes, I think they don't have loop of Henle, I suppose. Whereas in the places like in the deserts, like they have to conserve water, like desert animals, so they have like a longer loop of Henle because they need to conserve water. So the ability of the kidney to concentrate urine, it depends on the length of the loop of Henle. So as I mentioned before, like we have descending loop and ascending loop. And the important thing is the permeability, the descending loop and the ascending loop, they differ in how they are permeable to water. Okay. So the descending loop is permeable to water, and this water absorption or reabsorption, it's not it's a passive, so they don't need a lot of mitochondria or anything. So this descending loop, it doesn't have too much mitochondria. So and then the ascending loop, it is impermeable to water. That means it's there is no water exchange here. Whereas the lot of solutes, it is permeable to solutes. So a lot of like all these solutes comes out, and then the hydrogen also goes in for the acid-base balance. And this happens against the concentration gradient, and so they have like have a lot of mitochondria. And this permeability, the differences in the permeability between the descending loop and ascending loop, this results in the osmotic gradient in the interstitial fluid, and also it causes this countercurrent multiplier system, which is essential for urine concentration. You will learn more about this countercurrent multiplier system in the physiology presentation, how that happens, like, how these the osmolarity changes during, when the filtrate is going from the descending to the ascending. And the next thing is, is a distal nephron here. So this is a distal convoluted tubule and the connecting tubule and the collecting duct. And here, the distal convoluted tubule again have a role in water reabsorption and also the concentrating the renal filtrate. And around 5% of water is reabsorbed, whereas in the proximal tubule, around 66% to 70% of water is reabsorbed. Whereas here it's only less amount. And the important thing that you can see here is they don't have this brush border. They have the microvilli, but there is no brush border. But they do have a lot of mitochondria. And cuboidal, highly enriched mitochondria, lack brush border. So the macula densa, it's a part of the distal convoluted tubule, as I mentioned here before. So they are the specialized cells of the distal convoluted tubule. And they are tall epithelial cells, like you see here, and they're densely packed. And the part of the distal convoluted tubule which touches the afferent arteriole or passes the afferent arteriole, that region is called as macula densa. And these macula densa, they detect the fluctuation in the solute concentration. That is, whenever there is a decrease in chloride ion, then they communicate to the extra glomerular mesangium and also the renin cells, which are present next to this afferent arteriole. And thereby, if there is low salt, then the renin is stimulated. Okay. And thereby it regulates the renin-angiotensin system. So this communication between the macula densa and the JG cells, and thereby the renin, RAS regulation, this is called as a tubular glomerular feedback. And it's very, very important in regulating the blood pressure based on the solute concentration in the ultrafiltrate. So the last part where the filtrate goes for further concentration is the collecting duct. And here again, some amount of water and the sodium reabsorb. And again, based on where they are present, whether if they are in the cortex, then they are cortical collecting duct, and if it is in the medulla, then it's called as the medullary collecting duct. And this cortical collecting duct, they have two different types of cells, the principal cells and the intercalated cells. And if you see these two cells, they look different, right? The intercalated cells are dark because they are more cytoplasmic enriched compared to the principal cells. And And the principal cells, again, as I said, with the cortical collecting duct, there is some amount of water and the sodium reabsorption. And here around 8 to 10% of water is reabsorbed in the cortical collecting duct. And the principal cells do this water reabsorption under the influence of the antidiuretic hormone, and which helps in for aquaporin channel to present on the apical surface of the collecting duct, and thereby the water reabsorption occurs. Okay. Yeah. So and then the sodium reabsorption occurs through the ENaC, and the ENaC responds to aldosterone, and thereby it's also this sodium reabsorption also regulates the blood pressure through the renin-angiotensin system. And in addition to the water absorption and also the sodium absorption. Like there is also like a bicarbonate and hydrogen exchange, which is also like for acidification, acid-base balance. Yeah. Next, we saw the intercalated cells. The intercalated cells, the important role is for the acid-base balance, where there is some amount of reabsorption of the bicarbonate with the secretion of hydrogen going in. And the intercalated cells also like play a role in a little bit of water absorption also. Yes, like but the main role is for the acid-base transport, where the hydrogen goes in and the bicarbonate comes out. So then the last part is the medullary collecting duct. Okay, so the medullary collecting duct, the cells are called as IMCD, or the intermedullary collecting duct cells. So the urea transport is a characteristic of the unique feature of this intermedullary collecting duct. And this is the only place where the urea is reabsorbed and it goes to the interstitial fluid and raising the osmolarity of the interstitial fluid. And again, to some extent of water reabsorption occurs in this area also. And also like the hydrogen bicarbonate, that exchange also happens in this region. So this is important for the acidification of the urine. The final urine is at the pH of 6, and this bicarbonate hydrogen ion exchange helps for that. And here also the water reabsorption occurs under the influence of the antidiuretic hormone, which presents the expression of the aquaporin channels. Okay. Yeah. So now we've gone through like how the urine is formed, from starting from the filtration to the final urine, how it is concentrated. And this is important. It is important to know like the structure of kidney and all these different segments, how they have been concentrating the urine. Because if there are any defects in all these segments, they cause diseases. For example, like if there is a problem with the glomerular filtration membrane, that leads to the Alport syndrome and Goodpasture syndrome. And here they have this proteinuria because all these protein are excreted out in the urine. And the defects in the mesangial cells, it causes the nephropathy, IgA nephropathy, diabetic nephropathy. I think you'll listen more about all these diseases in the future lectures with the pediatric disease and other disease. And also like, there are several renal tubular syndrome, like the defects in different regions, like the proximal convoluted tubule and the distal convoluted tubule. They have like, it causes all these problems, like Fanconi syndrome. Yeah. Yeah, that's all I have for today's presentation. If you want to ask me any clarifications, yeah. So it's a brief overview of how the urine is formed and how the tubular epithelium, the collecting duct and the nephron tubular epithelium, they contribute in concentration. And in the physiology lecture, Lucas will go in detail how everything happens. For example, I was saying the counter current mechanism in the interstitium, how that salt gradient is maintained, how in the ultrafiltrate that regulates the GFR. Lucas will touch on those topics. Yeah. But if you have any questions, yeah. If I went fast at any of the slides, please let me know.